Welcome to the Performance Around the Clock podcast. I am Professor Satchin Panda, a scientist in the field of circadian rhythm. In this podcast, I will talk to experts in many different fields about science, health, and human performance. You'll also find out why circadian rhythms is my favorite topic. I hope you enjoy the podcast and thank you for listening. Welcome to another episode of Performance Around the Clock. And when I say performance around the clock, it's our biology around 24 hours a day, and also sometimes our biology changes over a month or in a season. And I'm super excited to have my longtime scientific hero, Charlotte Förster from Germany, who is also attending this Society for Research on Biological Rhythm conference in 2024 at San Juan, Puerto Rico. And she has some amazing stories on her personal journey through science that's very valuable for all of us, including colleagues, trainees, and everybody else, to understand what drives us to make very fundamental discoveries that has huge impact on human health. Sometimes those discoveries may not happen in human first, but they go back to very basic discoveries in, say, fruit flies, and we'll dive deep. So welcome, Charlotte. I'm
Thank
super
you.
excited. So every scientist has his or her own journey, how they got into science and how they get into what they're doing now.
Okay.
I'm always curious. How did you get into circadian rhythm research?
Perhaps to start, how I got to science. I was
Yes.
fascinated by science from the beginning. So natural science was really my favorite subjects at
Yeah.
school, and especially biology, but also chemistry and physics.
Yeah.
And so I decided to study biology. And I had to do it at the university that was closest to my home because my parents had to pay. So
Yeah.
I should stay at home and drive every day.
Which city was that?
That was a small, very small village, I have to say,
Yeah.
close to Stuttgart.
Yeah.
So Stuttgart was the university I had to go. And so I went to Stuttgart, and actually the teaching there was quite well, but I realized very early that I could not choose my favorite subject at that time, and that was actually neuroetology. So I wanted to study behavior in mammals and how it's controlled by the brain.
I see.
Yes.
So neuroethology.
Yes.
So how behaviors are controlled by brain.
Yes.
Yeah. And what did you do then?
Yeah, after the first semester already,
Yeah.
I changed to the University of Tübingen because there is a very broad field, so you could study almost everything. So I went there, and then
How far was that from home?
that was, I think, for American distances, nothing, but for German distances, it's about 50 kilometers,
See.
and there is no, how to say, not an easy way to commute.
I see.
And therefore, I had to have a small room there, to rent a room. And my parents finally agreed. So I was living in Tübingen.
Yeah.
Yes. And there I studied actually everything, and it was very interesting, from microbiology to genetics to animal physiology, but also plant physiology, biochemistry. So, and then already in the second semester, I listened a lecture from Wolfgang Engemann about circadian rhythms. And he was giving each semester a different lecture so that it was not boring for him. So
Yeah.
really prepared all chronobiology, from plants to animals to unicellulars, everything. So I was then listening his lectures every semester, of course studying the other things too.
Yeah.
And finally, yes, I asked whether I can perhaps do my diploma work in his lab.
So what is diploma work in?
Yes, that is a German thing. So nowadays we have also bachelor and masters. But before we had a kind of pre-diploma after 4 semesters, and then the diploma after 8 semesters. And when you do your diploma work, so it's a scientific work, like for the master work, and afterwards you are in principle ready, but you can start your PhD.
Yeah. Oh, I see. So it's almost equivalent to doing a master, so that you
almost
can.
equivalent, just that it takes, I think also from the time it's not so different.
Yeah.
Yeah.
Okay, so what was your diploma work?
Yeah, my diploma. Actually, I started in plants because I was very fascinated by the movements of the petal flowers, but also of the leaves.
Yeah, so they
Yeah,
do this
they
every
move
24 hours.
every 24 hours. So the petals close at night, open in the morning, or others perhaps even in the middle of the day, then close again. So that was really fascinating. And Wolfgang Engermann's main interest was to find out about the mechanisms of the clock. How is the clock ticking? And at that time, one did not know anything.
Yeah.
So the clock was regarded as a black box.
Yeah.
So you could manipulate the input and then observe the output. And the output are, for example, the pedal movements. And to manipulate the input, you could just add some chemicals to the water. So, for example, he did this with lithium.
Oh, I see.
Lithium was known as a medication for psychiatric disorders, especially for the bipolar depression. And it was known it lengthens the free running period of clocks.
In plants?
In plants too.
Oh, so plants also respond to lithium. I did not know that.
Yes, they do. And also to other chemicals.
Yeah.
So he just added the chemicals and then he guessed about the mechanisms in the black box.
I see.
Yeah. And when I asked him for a topic for my diploma, he also offered me, of course, many plant topics. But among the topics, there was one on fruit flies.
So this was a lab that was working on plants, fruit flies, almost like a zoo.
He had a zoo, yes.
And a botanical garden.
The botanical garden. And hamsters. We had hamsters.
I see.
We had these Tamius striatus. What is a kind chipmunks, but differently.
Oh, I see.
So different mammals.
Yeah. So it's very rare these days. I don't think there is any lab that can work on all these animals and plants at the same time.
Yeah, and also fungi, Neurospora crassa.
Wow.
And unicellulars.
So you had first-hand experience of looking at observing these circadian rhythms in all these organisms. I don't think there is anyone right now, any trainee that has that.
Yeah, it was really, I was very lucky.
Yeah.
I was also very lucky to learn basic chronobiology. And I have also to say at that time, Erwin Bünning, who is one of the three fathers of chronobiology, was still around.
Yeah.
So I had even a possibility to talk to him.
I see. This was in the 80s or 70s?
Yes, that was in 80s.
80s, yeah. Okay, so then you got, what did he give
my
you?
topic. My topic that I chose was to find the location of the master circadian clock in the brain.
I see.
At that time, that was not known.
By that time, in mouse, they had figured out that it's SDN, suprachiasmatic nucleus, but in Drosophila, there was no idea.
It was not clear. It was clear.
But in Drosophila, by that time, they already had the period 1 mutant, period 0, so they had the mutations. Potentially genes came around that time.
No, not yet.
Not yet.
At that time, they had no clue
what we are doing. So at least they had the mutations, but they didn't know where the clock is. Whereas in mouse and hamster, they knew where the clock is, but they did not have any genes. So.
Yes, but I have to say, in cockroaches, it was known that the clock is close to the optic system of the cockroach, and that was work from Terry Page.
So cockroaches also have circadian rhythm. Are they night active or day?
Night active.
They're night active.
Yeah, and cockroaches we had also in the lab.
Okay, so he also had cockroaches, like experimental subject, not like roaches.
No, as experimental subject, but also in the basement, I have to say. They had too.
So in cockroaches, the eyes are pretty big. Also in fruit flies, the eyes are big. So people thought that the clock was
Yeah, I think it was not because of that, but when you think about the SCN, the SCN has very clear connections from the eyes
I see. Okay.
via the retinohypothalamic tract. So that is important. And then I don't think that animals are so different. Of course, they had the idea that also something close to the optic system is responsible for the rhythm in insects.
Yeah, that makes sense, that the eyes have to give input to the part of the brain that's closest to the eyes.
Exactly.
Okay. So then you started looking.
Yes, and so the big advantage of Drosophila was that there were mutants around. And one mutant that is called Sina oculis has no eyes and also no ocelli, because the flies have 3 small eyes on the top. So absolutely blind.
Absolutely blind,
And a previous PhD student had recorded these flies and found that some of them are arrhythmic under constant darkness, so they seem to have lost the clock. And then Wolfgang Engelmann thought, okay, that is fantastic. You first record the animals, and later you dissect or you cut the brain in pieces, and then you see which tissue is absent. Sounds very easy. So I did it. And the first disappointment was that all animals were rhythmic in my hands.
So how many animals? It must be dozens of.
No, at that time we could only record 32 at one time. I have to say it was
32.
old recording systems with these event recorders and the deflection of the pen.
Oh, so you have to actually see the pen deflecting.
Yes, and then we had a paper that was running slowly, and later we cut our 32 channels in small stripes, and then we glue it on the paper to have an ectogram.
And then you look at it, and then
exactly, you could every day look, oh, what is my fly doing? Is it fine? And yeah, but they all were rhythmic, so that was a little bit disappointing. But nevertheless, I learned how to dissect, how to cut fly brains, so embed them in paraffin and then do paraffin sections and do photographs and stain them and then to see how big the optic lobes are.
So fly brain is what, less than a millimeter?
About 1 millimeter.
1 millimeter, and then you are spending a lot of time under the microscope and cutting the brains.
Yes, to make this kind of correlation, and it turned out that always when the flies had no eyes, the optic lobes was small, but there was no difference in the individuals.
Okay. So there was a big disappointment then.
It was, but to be honest, not so much, because at that time I already got contact to another scientist, Karl Friedrich Fischbach, who worked at the University of Freiburg, and he had several other brain mutants.
I see.
Very specific, who lacked certain parts of the optic lobes. And he proposed me, just try them. And I did so. And that was then my PhD thesis.
I see. So, many years ago, I think it was Jeff Hall or somebody, I don't remember who it was, who was trying to explain how genetics works or all this stuff. Then he says that, okay, suppose say you are spying for VW, Volkswagen, and you're trying to figure out who makes the windscreen in BMW. So every morning you go and tie up the hand of a BMW worker as they're going into the plant. At the evening, you go and see what is wrong with the BMW car. And then one day you'll see the wheels are missing. So then you know that that guy is responsible for the wheels. And then one day the door is missing. And then one day you'll see the windscreen is missing. And then you know that guy you tied up in the morning was making. So you are essentially collecting all these mutants, trying to figure out whether these flies are losing their clock.
Indeed, yes.
And then what happened? So you must have collected a bunch of
but it was at that time not so many.
Not too many.
Nowadays we have many, many more. So I don't recall. I had perhaps 5 or 6 different.
flies.
But I always characterized the brain anatomy and the behavior. And at this time, everything was much slower. Because there were no real computers around, so everything was manually. So you cannot just have, now we record thousands of flies at the same time, and
So it was very labor intensive work.
Yeah.
So anything came out of those screens?
Yes, indeed. So I realized about was one mutant that had really tiny optic lobes of about 5% of the normal size. And this mutant had only a certain type of cells of the medulla. The medulla is one part of the optic lobe. And this mutant had a crazy rhythm. So it was not completely arrhythmic, but it showed a kind of internal desynchronization in two components.
So that means every day it would wake up, say earlier and earlier, but at the same time,
Later and later.
after the nap, it would go back to sleep later and later.
Yes, indeed. So there were two free-running rhythms at the same time. And one had a period of about 22 hours and the other of about 25 hours. So you can really see the crisscross. And this I found very fascinating. And the only cells in this mutant were so-called medulla tangentials from the optic lobe. Of course, in the brain was also a little bit smaller, but And then I thought, okay, these are the candidates that perhaps somehow make a coupling between two different clocks. And if they are disturbed in some way because there are no optic lobes, then perhaps you can explain this. But the problem was that I had to find the identity of these cells and how they are changed in the mutants. So I was starting to look for any staining that could specifically label these cells. And at the same time, or a little bit earlier, Konopka and Handler published a paper where they could restore the rhythmicity of an arrhythmic per0 mutant by transplanting the brain of a per-short mutant into the abdomen of the per0.
Wow, so they took two different types of flies. Per0 is the flies that don't have any rhythm. They go to sleep and wake up in every 3 hours, 2 hours. So they took the head of the normal fly. Brain. So this is a brain transplant experiment. So they took the brain and removed the brain of the...
No, they left the brain in because the fly will not move anymore.
Yes. And then they put the brain inside the abdomen, inside the belly.
In the belly.
Inside the belly of this arrhythmic fly. And this normal brain, even in the belly, could actually make the fly go back to sleep and wake up every...
Yes, and it had a short period.
Wow.
That was important.
So that was I think that was the first brain transplant, successful brain transplant experiment. Whole brain in the belly.
Exactly. So it means that there is a transfusible substance coming from the abdomen, going to the brain head, and then somehow
making the fly sleep and wake up. So that seems simple, but the implications are big, that the brain doesn't have to really be connected.
Indeed.
But it has to just secrete a signal that can travel and then reach.
But it is somehow analog to the experiments that Ray Silver carried out. Transplanting the suprachiasmatic nucleus in an arrhythmic mouse. And then packing it in a kind of sac that is, diffusible, but no nerves can grow out. And then she could also show that the rhythm come back.
So it was very similar. But Ray's experiment was around the same time or different time?
Oh, I don't remember. I think it was a little bit later.
I thought it was a little bit later.
So the Handler... I don't remember.
Because Ron Konopka must have done these experiments before mid-80s. Okay, so now you had the idea that there is some neurons or the brain secreting something.
Indeed. And there are neurosecretory cells. So I think they are homolog, but let's be careful to say analog to these neurosecretory cells that secrete into the pituitary.
Yeah.
So flies have something very similar.
Yeah.
And I thought, okay, that must be these neurosecretory cells.
Yeah.
And I was looking for stain to stain them. But how to do that? So another perhaps
So you're still doing your
at the Botanical Institute in Tübingen. Still, I did all that.
But this was still you were finishing your project,
That
master's
was,
project. Or no?
I have to say, that was still during my PhD.
Oh, so you started your PhD after you finished the.
Oh yes, I forgot to tell you. So after I had this disappointment
Yeah.
with the first mutants,
Yeah.
I had my diploma,
Yeah.
but then I wanted to go on with a PhD and find out whether the other mutations perhaps have a disturbance.
So that must have been a turning point because you had a disappointment.
Yeah.
That whatever you tried did not work, but still
I had a new idea.
You wanted to pursue this new
Yeah.
idea because a lot of people would give up right there and then do something else. So that was you are resilient. You really wanted to pursue this question.
Yeah, because I saw a way.
Yeah.
I failed the one way, but there must be another way to do it.
Yeah. Okay, so this was during your PhD. You figured out that you got the idea that it must be neurosecretory cell.
Yes, that was during my PhD, and at the end of my PhD, I tried to stain the cells. So that was really the end of the PhD.
Yeah.
And we had a visitor from Warsaw in Poland.
Yeah.
And that was something else. So we tried to find the clock also in a larger fly, Musca domestica.
Okay.
And this guy, Bronislaw Szymborowski, he was very skilled to do dissections in insects.
Oh.
So to cut the optic tract.
Mm-hmm.
So the connection between the eye and the brain.
Yes.
So we decided to do that. So
How big are these flies? These insects?
They are
5 millimeter, 10 millimeter?
5 to 10 millimeters. Yes,
yeah.
Depending.
So still, it's huge compared to fruit fly.
Huge, absolutely huge.
Yeah.
So we really cut this and sealed it, and then recorded activity, and then looked whether they become arrhythmic or not, and later we stained them to see where we had made the cut.
Yeah.
Okay, and this guy could also stain neurosecretory cells.
I see.
And then I visited Warsaw for 2 months to learn the technique.
Yeah.
And then I came back to Tübingen and indeed found that these neurosecretory cells that I had in mind, that are close to the optic lobe, are still present in my mutants that still showed the rhythm, but with internal desynchronization. And that was, of course, very important.
Only when you cut the connection, you saw this internal desynchronization?
Only in these mutants.
Only in these mutants. Okay.
In the flies, we cut the connections. And we realized some of them are arrhythmic and some are rhythmic. And then we concluded that the master clock must be just at the border between the optic lobe and the brain. And actually, that's true. But the only thing what we could do there.
Wow. So this is almost 3 to 4 years of PhD, 3 years.
Oh, I needed only 3 years.
3 years, yeah. In Germany, it's 3 years after you have done your diploma, right?
Yeah, actually, rarely people can do it in 3 years. Most need 4 years, at least.
Yeah, but it's a lot of very groundwork, trying different mutants, going and learning dissection and surgery, and then testing them again. Yeah. So it was the end of your PhD then. You knew that it's close to the optic lobe, but
I knew that it's close to the optic lobe. Yeah, and then I was pregnant and had my first child. I have to say, and that was at the beginning a stop of my career. But
I see.
I wanted to continue, and after my child was 1 year old, I tried to continue and really find out what are the cells. And I was lucky because for my PhD work,
you got your PhD degree before you became.
So I hurried up a little bit, so I got it just 2 months before the birth of my child.
I see. Okay. You waited for a year and then you came back to look for this.
Yes. Yes, exactly. For my PhD, I got an award. And that was important because it was 5,000 Deutsche Mark. And that allowed me to travel. And so I visited each chronobiological conference that I could,
Yeah.
just to learn more and to meet people.
So you still have this 1-year-old child.
Yes, it always was a problem,
Yeah.
but my parents had to come to care about the child.
And then
So
you
I
could
had to
go.
be very efficient in doing everything.
Yeah. So you went to all these conferences.
I went to the conferences, and I went especially to one conference, the Gordon Research Conference.
Mhm.
And here I met Monika Stengel, who was working in cockroaches and in crickets, and she had a poster
Mhm.
because I was looking for an antibody that might stain the cells that I had discovered. And this antibody staining she had was from the pigment dispersing factor
Mhm.
and cockroaches and in crickets. And I saw the poster and was immediately convinced these are the cells that I have to stain for.
Wow.
So we started a cooperation to stain with
Yeah.
the antibody. I had first to learn this antibody staining, I have
Yeah.
to say,
Yeah.
and then to apply it.
So I'm just visualizing this, what is happening. So you had a 1-year-old child.
Mhm.
You're leaving her or his. This is son or daughter?
A son.
Son. You're leaving him with your parents, and you're on a global hunt to look for the which staining may stain these cells, right? You're going from poster to poster in every conference.
Yeah, come on.
Kind
I
of
was
like
much
that.
more selective, so I only went to the
Yeah. You are not going to all conferences, but only chronobiology, circadian
Yeah.
conferences.
Indeed.
Yeah. And then you are very selective that you want to look for the insects
I wanted,
posters.
yes, exactly.
Yeah.
Yeah. But there was another reason to go to this conference because also Michael Rosberg and Jeff Hall,
Yeah.
and I think also Michael Young were there.
Yeah.
And at that time, they had an antibody against the period protein, so one of his main clock proteins.
Yeah. So let's connect them back to the story that Jeff Hall, Michael Rosbash, and Mike Young, they were working on that time to clone this period gene whose mutations make the circadian clock arrhythmic or running short or running faster and slower. And all three of them, at that time, they were the only ones who actually had a mutation that affects behavior in any animal. And so you also were very strategic to make sure that if they're looking for the gene and you're looking for the cell, then maybe if they have any clue, then you can get those clues.
Yes.
To some extent.
Yes, I wanted to know whether the cells I had in mind were really clock cells, so I needed to show this.
So they had the mutation. They did not know where the cells are, and you're looking for the cells.
Yes. That was the main aim.
Yeah. And then you met them.
Yes, I got the transgenic line, not the antibody. Because
So let's go back. So you met Jeff Hall or Mike Young or Michael Rosbash, and then.
So I was asking both Michael Rosbash and Jeff Hall whether they could give me a little bit of antibody, the anti-period antibody to stain. And then Jeff Hall was the one who answered. And he said, yes, of course.
Yeah.
But I recommend you to use a transgenic line because this is much more reliable than the antibody.
I see.
And then I said, okay, yes, of course. Thank you very much. Would you send it? And he did.
Wow. So he sent it from US to Germany.
Yes.
That fly. And some of these connections are very important because only because he sent you that fly line that changed the story.
Yes. Yeah, he was very generous.
So that means for you to come to that conference and to see this clue from cockroach or
cockroach and cricket.
And cricket, and then you're making all these connections. It's almost like a murder mystery. You're solving a mystery. So you went back, but by this time you could work back in the same lab, or how was the
the situation was that I was not paid for anything, but Wolfgang Engelmann was generous that I could use the lab. So whenever there was any possibility, and my child was in care, not so easy in Germany, but then I could indeed use the lab and do the staining. And that is what exactly I did.
So you were unpaired, but you still had this drive to discover the cells.
Yes. A little bit later, I got a small stipend, but that was not much. That was 200 mark per month.
Yeah.
But at least I had a little bit.
Okay.
And I could buy some chemicals and also, literature, books also.
Yeah.
And the forceps to dissect the brains.
Yeah, but no salary.
No salary.
You got the stipend and you spent on books,
Yeah.
forceps, chemicals. Okay, so let's continue. And then
Yeah, then I did it.
yeah.
And my idea was right.
Mm-hmm.
So I did it in cooperation with Uwe Homberg.
Mm-hmm.
And we stained the different mutants and the wild type with this antibody.
Yeah.
And I realized, yes, there are these cells, neurosecretory cells, that are at the same time medulla tangentials.
Mm-hmm.
4 cells each brain hemispheres.
So 4 cells, they constitute the master clock.
Yeah, not really, because there were 4 smaller ones at the same time.
Okay,
and actually
so 8.
they, I think, have the master clock, but they also stained with this antibody. And in the mutants, they were still present. But in the mutants that showed this internal desynchronization, so the 2 components, the pattern of these arborizations of the fibers were completely crazy. So they run over the brain everywhere.
I see.
So for me, very clear that the communication within the clock is disturbed. Meaning also that this pigment dispersing factor must be the one that is important for communication between different clock neurons.
I see.
But at that time, we did not know what are the different clock neurons. So that came a little bit later, but in the lab of Jeff Hall.
Yeah. So pigment dispersing factor, let's also spend a couple of minutes on that. So
okay,
how did it come from which animal? Because one thing is, in biology, we often get clues from here and there. But the pigment dispersing factor was not originally discovered in flies.
No.
Yeah.
Actually, it's the pigment dispersing hormone.
I see.
And that is present in crabs, in crustacea.
Crabs.
In crabs, yes.
What do they do in crabs?
Crabs can change their body color.
Yeah.
And they also can adapt to light by dispersing a red screening pigment in their eyes to distally.
So it's their sunglasses.
They have kind of sunglasses, indeed. And for this kind of sunglasses, you need the pigment dispersing hormone, because the pigment dispersing hormone makes these pigments migrate to the margin of the eyes and shield them from the light.
Yeah.
And then during the night, you need a different hormone, that is a red concentrating hormone, and then the pigments migrate back so that the eyes are not anymore shielded and can see more.
Yeah. So this is beautiful, that something that was discovered in crab, you're trying that to see whether that hormone or something similar to that is also present in the master clock neuron, potentially master clock neurons of Drosophila.
And it was discovered in the cockroach by Monika Stengel and Uwe Homberg.
Yeah.
And they already did know that there is not a rhythmical migration of screening pigments, but it has another function in interneurons. So they were the first with pigment dispersing hormone, I have to say. But I then could show that it's also present in the fruit fly and that it is different in mutants.
I see. Yeah. So that must have been a very exciting moment, that finally, after so many years, starting from your undergraduate research, you have been looking for these cells, and you completed your PhD and then went on an international hunt for this gene and came back.
Yes, and I was able to show that indeed these cells are clock neurons, so they express the period gene or protein.
Yeah.
And I was so excited that I thought I should send it to Science or to Nature, but of course it was not taken.
Yeah.
But fortunately, proceedings of the National Academy of Sciences took it.
Yeah.
So it was a long procedure. Yeah. For me, too long, because at the same time I applied now for a real stipend where I got payment.
Yeah.
And
So until now, until this discovery, you are actually doing all of this unpaired.
yes.
Wow.
And I did know, but I need the payment to continue. So that was really the moment that
Yeah.
either I get it or I'm out.
Yeah. I see.
And I did not get it. And
Oh,
then I was really completely disappointed.
you did not get this stipend even after publishing this?
No, because it took too long.
I
You know,
see.
sometimes very crazy things happen.
Yeah.
So at the Botanical Institute, I was a nothing.
Yeah.
And so when I handed the paper, at that time, we have to send the manuscript, 4 copies,
Yeah.
such a package, to US.
Yeah.
There are 2 ways, by
Yeah.
flight or by ship.
Mm-hmm.
By flight costs a lot of money. And so for me, it was clear it has to be sent by flight.
Yeah.
But the secretary of the institute decided for her, we will not spend the money. And she sent it by ship.
Oh,
So it took 4 weeks until it finally arrived.
wow.
So meaning that everything was delayed and it
Yeah.
was, of course, not published. So
Yeah.
it was still unpublished.
I see.
And that's the reason why I did not get my grant.
Oh,
Yeah, crazy, but it was like that.
then did you get the grant? How did you get back to actually getting paid?
I started the paid job at the Max Planck Institute and completely different things. So
But how were you supporting yourself during that time when you were?
I was married and my husband had a job and my task was to look after the children.
Okay.
Yeah. So it's a classical model. At that time, it was completely normal in Germany. So everything what I did on my own for myself, that was voluntary.
Oh,
But it was expected that I do the household and care for the children. So of course I did that too.
so you were doing 2 full-time jobs
Yes.
and not getting paid for the second full-time job.
That's true. Yeah, but it's good to have friends.
Yeah.
So first of all, they helped me to get these 3 months contract at the Max Planck Institute to do something else.
Yeah.
So that was good. Finally, I had some payment.
Yeah.
And this contract was then prolonged and prolonged.
Yeah.
And then
This was Max Planck Tübingen?
in Tübingen, yes.
Yeah, Tübingen.
Max Planck Institute for Cybernetics.
But it's not circadian. So what are you
No,
doing?
it was not circadian. No,
Okay.
no, I did recordings of the neuronal activity in brain slices of guinea pigs to find out about the effects of anesthetics. So that was a project of somebody else. So I was just paid to do it like a technician, but it was okay.
Yeah.
Yeah, it was quite okay.
Yeah.
But of course, I wanted to be back in my field.
Mm-hmm.
And now I got help from Jeff Hall because he heard that I was successful to do this co-labeling with the transgenic flies he sent me and this pigment dispersing factor antibody.
Yeah.
And he invited me to Brandeis. And when I traveled there, I told him the entire story. And he then wrote a letter to the German Research Foundation that it was not fair not to give me the grant. So they couldn't give me then the grant, but they gave me a smaller grant for 2 years.
Yeah.
So that was fine. So he really helped me.
Yeah.
So I finally could go on.
So his letter was really crucial for you to get this 2-year grant, to do what you wanted to do. Means you were getting paid to do some recording from guinea pig brain, but that's not your passion.
It was also interesting. Perhaps it could have been my passion, but you know, I first wanted to finish the other main interest.
Yeah. But this 2 years of fellowship or stipend, did it come with your salary?
Yes, I had my salary and I had very, very little research money, but it was enough to continue at least.
So that was the first time you actually
were paid for my...
were paid beyond this 3 months contract.
Yes, indeed.
Yeah.
That's true.
Yeah. You stayed with the university there?
I went back.
Yeah.
Wolfgang Ellmann was still there and he offered me the space, so I could work in his lab for the first time. Yes,
So this is also very important for the mentor to give you this opportunity to continue, although he could not support your salary from
Yeah, but he was a very good mentor, I have to say.
Yeah.
And by the way, maybe you know, he is not on the papers.
Oh,
He said it's your work.
yeah.
I can't contribute anything. It's enough when you mention my name in the acknowledgements.
Oh, that's so generous of him to do that. Yeah. Okay, so then you continued with means. I think the first time I got to read your paper was when you had done the glass mutant work to show that the photoreceptor. I think that time people are still looking for what light receptor entrains the.
Yeah, sure. We were very much interested in the light also because of these blind mutants that have no eyes, and they still could perfectly synchronize the rhythm to the light-dark cycle. So there must be an additional photopigment, something. Yes, it's true. You know, it all goes back to the cooperation with Jeff Hall that started after I visited him. The first thing what we did is a cooperation with Maki Kaneko, because Jeff wanted to know about the clock in larvae, and they were not able to find it, to stain it. And I told him, yeah, but it's no problem. I have already seen the cells. They are there in larvae. And he then got completely excited. Yes, we have to do it. And so we started with cooperation, and then could also show how the circadian system in the fly slowly develops. So from the larvae, then in the pupae, until they are adult.
Yeah. So then how, when did you finally get your position as a faculty or as a researcher?
Okay,
What is the story?
I got a kind of position, but that was again a kind of fellowship for my habilitation. And there I moved to a different lab because Wolfgang Engelmann was now retired. So to the lab of Hans Erkert, who is also working in circadian rhythms, but in monkeys.
Okay, in monkeys?
In monkeys, yes.
In those days, okay.
In night active and day active monkeys.
Oh.
So it's another story, but he was a chronobiologist, and he understood that I wanted to do chronobiology. He gave me a tiny place in his lab where I could work. I was very thankful to do that. Okay, but for my career, then I did my habilitation. That is a German thing, that you show that you can teach and also publish. And after you get this habilitation, you are free to apply for a professorship.
So what did you teach? Chronobiology, of course?
No, because that's not a subject in Germany.
I see.
I think Teller Rönneberg is the first who really said he is a professor in chronobiology.
Really?
I never reached this state.
I see.
So I taught zoology at the University of Regensburg.
How far is that from?
About 300 kilometers.
Oh, so you had to move to a very different city and.
Yeah, but I commuted because my family stayed in Tübingen.
So you went for the week and then came back in the weekend, 300 kilometers.
Yeah.
And you still have these kids that are growing up.
Yeah, but they were now teenagers.
Okay, so they were for your habilitation, sorry. For your professorship. So
yes, and I had the first professorship, so that is a real paid position at the University of Regensburg.
May I ask, how old were your kids by then?
My kids were 13 and 15, as far as
so it's almost 15 to 17 years after your PhD.
Yeah. I had my PhD in 85.
Yeah.
I had my habilitation in 2000. Because that was all this period.
15 years.
And I started in 2001 the professorship in Regensburg. A long time.
Yeah. So since that time, you have done a lot of work in Drosophila
and other insects.
But in the last few years, you have been giving these very illuminating talks about lunar rhythms. So you were interested in circadian rhythm, but I guess your overall interest in the biology of time.
That's true.
What are the other timing that interests you? And then how did
Of
you
course,
get
seasonal timing.
seasonal?
It's very interesting.
Yeah.
And the long-lasting question whether the circadian clock is involved as a time reference for measuring day lengths. That
Yeah.
was something I really would like to show. And now we are working on this on different Drosophila species.
So do Drosophilas have seasonal rhythms?
Not Drosophila melanogaster. Yes, they have. I have to be careful. My colleagues will kill me if I say not. No, they have a reproductive dormancy.
I see.
But they need short day lengths and a low temperature to do so. It's not completely alone photoperiodic.
Yeah.
But when you go to the very north,
Yeah.
there are northern Drosophila species, Drosophila isoana, Drosophila littoralis, Drosophila montana, and they have a real diapause. So that is obligatory. And when you shorten the day lengths, they immediately stop to reproduce and go into this hibernation state.
So in wintertime, Drosophilas don't reproduce.
No, they can't. No.
They can't reproduce or they don't reproduce. So this work relates to a lot of things that we experience. For example, mosquitoes. They're very seasonal. They reproduce and expand in certain season, and then they disappear. We think that the temperature changed, they disappear, but actually it's the biology, the circadian clock that may be interacting with temperature and how short the day length is, and that's how they're determining when to reproduce. Yeah. So
yeah, that's true.
Yeah. So you were working on the seasonal rhythms.
Yes.
But what is interesting is in the seasonal rhythms, you're bringing up two aspects. One is the day length has to be short, and the temperature has to go down. So two things have to coincide for this trigger to see some of the seasonal rhythms. Not all seasonal rhythms are like that, because, for example, in plants, you can just change the day length and it will trigger the plants to flower or pause for flower.
Yes, but to be honest, in these northern Drosophila species, it's exactly the same.
Really?
Because for them, it's so crucial to go to hibernation in time that they don't rely on temperature. So you can even induce it at 15, 20 degrees.
Really?
Yeah.
Wow.
In the lab. So we made a very crazy mistake when we got the flies and cultured them. So usually we have a 12 to 12 light-dark cycle for our Drosophila melanogaster come from the tropics.
Yeah.
And we put the flies there and they always died. Until we realized they need a long day, at least 20 hours or 22 hours
of
light. Yes.
Really?
Yeah. Or
So
constant light.
These are the Nordic flies.
Yeah, they are Nordic flies. So if you don't have them under these conditions, they die.
Wow.
So they don't die, but they go into reproductive diapause and have no offspring.
I see.
Yeah,
I had no idea about that.
we also at the beginning, finally, we thought,
Yeah.
You have to have them under constant light.
Yeah. So is there anything now? So you're working on how flies integrate this day length and then the clock to decide when to reproduce.
Yeah, more or less.
More or less.
More or less.
Yeah. But now in this conference, and also I think I heard you in European Biological Rhythm Society in Zurich, you talked about another rhythm, lunar rhythm.
Yeah, but not in flies.
Not in flies, but I want the listeners to hear about how you got interested in it, because there are many stories, just like this story that you just mentioned. Those are never written in any manuscripts. We'll never hear in any scientific presentations.
Okay. Of course, I did know that especially marine organisms time their reproduction according to the moon, because they have to meet mostly once a year in a certain moon phase.
Yeah.
And even terrestrial animals often have reproduction according to these lunar months. And since females also have a reproduction cycle, so a menstrual cycle about 30 days, then I always thought that is not just by chance. It's probably that perhaps in former times, when we were much more aware of the moonlight and the natural, yes, exactly, that it was indeed synchronized. So that was my idea. But how I came to it, My mother taught me that I always have to note the first day of my menstruation in a kind of calendar, just when I have to visit the doctor for any medical reasons. I have to know it. And I just did it. And I did it for my entire life. And I did not throw the calendars away because other important notes were in the calendars. And then I noticed when I was...
So you kept all these calendars for...
Yes. Small ones.
Oh, small ones, not the big ones.
No. Very small ones. I just kept them somewhere.
Yeah.
And when I commuted between Tübingen and Regensburg, so I traveled always late at night.
Okay.
And then I was very aware of the moon, because there's a big difference when you have to walk in complete darkness or in the moonlight.
So where? So in Tübingen you had to walk, or in both stations?
I had to walk to the station.
Okay, so walking from the lab to the station or the apartment to the station, yeah.
Yes. And then I noticed that my own cycle was absolutely in synchrony with full moon.
Wow.
For at least one entire year.
Did it just strike you right away, or after a few cycles? Yeah.
It struck me right away. And then I noticed after some time it was not anymore. So it was not synchronized anymore. And when I had a little bit of time...
So you're still commuting, but it was not synchronized?
Yes, it was about that time. Yes, it stopped synchrony.
After you stopped commuting?
No, during the time. So it had nothing to do with that.
Okay.
It was just by incident,
by chance,
that I noticed that it was synchronized.
Yeah.
Okay, I had some time, and then I plotted my cycle from the calendar together with the moon cycle to see about this synchronization.
Yeah.
And then I realized that before I was synchronized to the full moon, and actually I was 3 years synchronized to the full moon, before I was more than 1 year synchronized to the new moon.
Wow.
But I never noticed.
Yeah. Of course, you will notice with the new moon. Full moon is noticeable.
Yes, but not the new moon.
Yeah.
Okay, so I saw many interesting things in my own cycle, and that I could not explain by chance. So there must be a kind of entrainment or synchronization to the moon. And then I got very excited, and I wanted to have more data from other women.
Yeah.
So then from that time on, I asked every friend, colleague, whatever, whether by chance they had noted their menstrual cycle in the calendar.
Yeah.
And not many had. But some had.
Yeah.
And so after...
But at that time, let's dial back, means you're just inquiring, and there is no ethics committee review or anything, because you're actually not asking for the medical records or anything, and you're not changing any health behavior, you're not giving them medication, nothing. So it was perfectly
I only wanted, and they give it all voluntarily. So it's...
voluntarily they're giving you this. Yeah.
And of course, I anonymized the data, so it's very clear. And then after about 10 years, after 10 years time...
After 10 years?
I had the cycles of 22 women.
So you continued asking
but that was not my main work.
No, I mean, it's on the back of your mind, and once in a while you might meet a friend or colleague with whom you'll feel comfortable enough, because you won't just go around and ask for this information. So it took you a really long time to get 10 or 20.
It's not easy to find so many crazy women who know your cycle.
Maybe these days with the apps it's okay, but then in those days when there was nothing. Yeah.
Yeah, okay.
So it took you 10 years to get 20 plus records.
Yes.
And these records are more than a year.
Yeah. So the shortest that I took were at least 5 years.
Oh, so you have to find really crazy women who... 5 years.
And the longest at that time was...
Not only they have to note, they have to actually give you physically the calendar or some written record.
They can photograph it or copy it, of course. Yes, and most it was just crosses in the calendar, and they copied it and sent me the paper that it was.
Yeah. Okay, so after 10 years, what did you find from this 22?
I found that I was no exception, but that also the other women temporarily were synchronized to the moon. So never ever for the entire life,
Yeah.
but only for certain months,
Yeah.
years. It was completely different in the different women.
Any chance you correlated that to whether they were urban or rural dwelling? Or...
A little bit.
Okay.
But, with 22 women, you
Yeah.
have such a small N that you have to
Yeah.
be very careful.
Very
But I
careful.
did, because I found 3 women who had an extremely short cycle.
Mhm.
So from the beginning, what I forgot to say. So it's very regular,
Mhm.
but the women start with a period that is not as long. So very often longer than 1 month.
Mhm. Yeah.
And then over life, it slowly shortens and shortens
Mhm.
up to 25 days.
Mhm. Yeah.
And so what I did want to say, yes, I found out that 3 of my women had an extremely short cycle from the beginning. So only 27 days cycle. It's quite short.
Yeah.
And all 3 were from really big cities.
Wow.
And they were all 3 evening types that exposed themselves a lot to light at night.
Yeah.
And one was even sleeping with a night light because she didn't want.
Yeah.
So, but we have to be careful. But I had the impression that light sometimes, speeds up the cycle.
Yeah.
And then it's not anymore synchronized.
I see. So that was in the back of your mind, because you know that circadian clocks
change their period under light. And this seems to be also the case for the menstrual cycle.
Yeah.
And it was published by Christian Kajochen, as far as I remember, for a completely different reason.
Yeah. Yeah, actually, I will see him in 3 weeks.
I can ask
He's
him.
also here.
He's here. I saw him.
Yeah. Yeah. So with these 22 records, when did you start presenting this data?
I think the first time I presented at the EBRS, it was already 2012, I think.
2012, yeah.
But at that time, it was very incomplete.
Yeah.
It was the first time.
Yeah.
And then at the Gordon,
Yeah.
more complete. When was it? You know it, I think.
I think I saw 2022. That time you had more data.
Yes.
Yeah.
Exactly. And in 2022, I just was able to publish it. So it was published in 21, in January.
You started
21.
collecting this data in 2002, 2003, something like that, right?
But it took me 20 years to publish. But it is my side project. It's just for fun. It has nothing.
It took you 20 years for this side project. So meanwhile, but you have collected now many records.
Yeah.
And you, in this conference, you presented that a lot of data are now coming from Italy, northern Italy. What's the story there? You became famous in northern Italy.
Yes. No, I have friends, colleagues from the University of Padua, and they were really fascinated by the study, and they wanted to contribute. Actually, we even wrote a grant together, a grant application, because Rudi Costa had the idea. He has also to study whether men have similar lunar rhythm, and perhaps they are more fertile at a certain moon phase. But that was part of the study. The other was just to enhance the study to different latitudes, at different light, so big cities, small cities, and so on. So the grant, we did not get the grant, but nevertheless, we started cooperation. And another colleague of Rudi, he is a medical doctor, and he had access, or he could ask easily women who send the data. So I have now many Italian women in my database.
Yeah. So you mentioned light can disturb the cycle, and you personally also observed only, of course, the first time you observed was when you were walking to the station on a full moon, then you made the connection. So now that you have collected all this data, and people are also collecting from smartphone, is this rhythm in menses still lunar? in modern days? What do you see from cell phone self-reported data?
What I see in my data that I now analyzed is that from 2010 onward, the rhythm, so the synchronization to the full and new moon is gone. So I could see it in single individuals, but overall it's not anymore significant.
So from 2010 onwards, you don't see that alignment of menses with the moon phase, whether it's new moon sometimes or the full moon.
Yeah.
And what's the hypothesis now?
Yeah. So first of all, why I say
why
from
2010?
2010.
Yeah.
So of course, we know that light pollution is increasing continuously.
Yeah.
However, at 2010, these light-emitting diodes came on the market and they replaced the common bulbs. And there was one paper showing that then the light emission from Earth, so they measured by satellites, was increasing like crazy.
Yeah.
So I decided to take this 2010 as a border to calculate the entrainment from the rhythms before and after. So that is, of course, a little bit arbitrary, but before it was synchronized to full and new moon temporarily. Yeah. But afterwards, I could not see it anymore on a significant level.
So you also have personal record from women, from the same women, before 2010 for a few years, and then after 2010 for a few years?
Yes, I have. Yeah. However, this is difficult because with age, the rhythm becomes shorter.
Yes,
and
with
so
age.
I cannot really draw conclusions from a single woman. I have some that are really synchronized at the beginning and not at the end, but then I cannot distinguish, is it the age or is it light pollution?
But after the paper was published, are more women coming forward and sending you their own record?
Yes, indeed. And therefore, I have now 157.
157 records. What is the longest and what is the shortest record?
Okay, the longest was 37 years.
37 years, almost the entire reproductive life.
The entire reproductive cycle.
Wow. That's... So you have really I think you may be the only one who has the longest record.
I assume so.
You must have collected. And this is a treasure trove because it also brings up this issue. So now going back to human health and other stuff, is there an advantage of being aligned to the moon phase? Because this is a huge question. We may not have an answer, but we may have some speculation. And I want to hear your speculation.
Indeed, I have no answer.
Yeah. It's good. Thank you. Because sometimes, Francis Crick was president of Salk, and then sometimes people would ask him questions. And then he was very humble. He would say, if he knew that there is an answer, but he doesn't remember, then he would say, I should know this, but I can't recall or I don't know.
No, I don't know. But I think it has some biological relevance. I'm quite convinced. Otherwise, this would not be conserved throughout the entire evolution of humans.
But you also mentioned that monkeys do have these rhythms.
Yes, some monkeys.
And also cows have these rhythms of...
Yes, they have a higher fertility at new moon and full moon, but I don't know why. It's just an observation.
So this is a phenomena found in monkeys, cows, and maybe in many other animals, but people haven't just monitored and reported. Yeah. So what is your hypothesis or opinion?
Okay, I think it must serve synchronization between females and males somehow. Yeah. And when I think of former times, really very, very way back, I believe that most of the time humans spend outward is to hunt and to collect. And I don't think that men and women could spend every night together. Perhaps the men were really hunting and outside and traveling or whatever, and then come back. And reproduction is so important that you should be back in time. And then it would really help to know that either full or new moon is a good time to be at home. So that was my very first idea. And the other idea that came up later, after I read a paper from Pecker et al., published 2011, about lion kills in Tanzania.
Yeah.
And that was an amazing study.
Yeah.
Because I think it was a period of about 10 years where we
Yeah.
recorded these lion kills, and they found out that lions hunt at the beginning of the night, so in the first half of the night. Just when dusk sets in,
Yeah.
they kill most animals, but
Yeah.
also sometimes people.
Humans, yeah.
Yes. And what I did, I took the data because it was exactly given when they recorded these kills. And I separated for waxing and waning moon.
Oh, okay.
Because I did now in waxing moon, the moon, you see it already in the afternoon, and it's up at the beginning of the night at dusk. So it's light, you can really see outside.
I see.
But during waning moon, the moon rises in the second half of the night. So it's really dark outside. Okay. And when I plot the data, then I see that there are significant more kills when people went out during waning moon. And during waxing moon, it was much, much less.
So
and the reason,
when there is light, then the lions don't come out? Or don't?
but the lions, they can see much better at very low light.
Yeah.
But humans don't see.
Yeah.
And then I think if somebody goes out, if it's really dark, the lion can still see, but you don't see the lion, and the risk to be killed is higher.
I see. Okay.
Lesson from that, don't leave your house during new moon or the waxing moon, the waning moon.
Yeah.
And then, of course, if you should not go out, perhaps it's better to stay home with your wife.
Yeah.
And the ones who follow the rule have more kids. So it's a very
I'm amazed, like how you pick these clues to solving this mystery from Tanzania lion kills to
But it's not proof.
It's not proven, but it's a good story, and it, sometimes we have some stories that make sense, but we also have to, like you said, like you clarified, it's not the reason. It may be one reason, or this makes sense. And you are very humble to say that it's not the mechanism, or this is not the reason why, but this is what it. So this is the second one. Do you have a third one? No,
actually
these are the
not.
two strong ones.
That are the strongest. But I would not completely exclude that some of the fertility issues that nowadays are really increasing are because we do not live anymore our natural life. Because we don't know what else is affected by this light pollution.
Yeah. But you also mentioned that you have access to this self-reported cell phone data on menses.
Yes. So I have to say the new data, for example, the data that the Italian colleague sent, were all collected by cell phone apps.
I see.
And of course, these women expose themselves much more to light, artificial light, and this blue light.
Yeah. So then you don't see that
could be that is also correlated. But we also spend so much money in front of television or other screens that could also completely confuse. So that is.
So Charlotte, what is new? What is keeping you awake at night these days? Anything exciting? Or this itself is a big mystery to solve?
I think we have enough to solve, and I got even at this conference new ideas what to do in seasonal adaptation, but also in the lunar clock, and also for my circadian clock in Drosophila. So there are so many exciting biological phenomena one can explore if one wants.
Yeah. Thank you so much for a lovely conversation, and I really got to know you way much more during our conversation. This is something that will never be published in any manuscript. You'll never talk about it in a conference, but this is very important to know how scientists go through their career, how they pick up clues here and there, and sometimes you need to have that patience of 10 years, 20 years to make those breakthroughs, because these are once in a lifetime kind of breakthroughs, and those are possible with this perseverance, resilience, and pure determination. Thank you, Saturday. I'm so glad and I'm honored to speak with you. Thank you.
You are most welcome. Thank you too.